All right, thanks everyone for being here. Next talk is on Gator Bio. My name is Sri. In the next 10 minutes, I want to show you how Gator Bio is addressing three of the most often mentioned issues that customers have in antibody discovery in conversations with us. Just to give you a brief introduction to Gator Bio, we are a company that commercializes Biolayer Interferometry systems for label-free binding analysis, something that our previous speaker talked about in terms of BLI, SPR, and other technologies being used commonly in the discovery screening workflow. BLI technology is a bit unique in that it has a wide variety of applications. You can use it in antibody discovery workflows early on, but also take it downstream to do quantitative measurements of antibody titer, other protein titer, AAV, and other virus particle titer in bioprocessing, cell line development workflows. So it has broad utility in drug discovery and development. And Gator Bio is, what we're trying to do is, the original inventors of BLI have started this company to really accelerate innovation in this space to problem solve the most interesting issues that customers are facing currently. So often we hear issues with, in general, right, across multiple platforms, variability and reproducibility. Issues can slow down the workflow, require repeat of experiments, and increase cost. Limitations in sample throughput often come into play because of the lack of flexibility of systems when you want to do different types of experiments in your experiments in your workflow. And then overall, there's just a rising cost with respect to instrumentation and consumables and services. So we want to address some of these pain points that you face in selecting the right technology for your workflow and utilizing them the right way in all the experiments. So we understand that you're selecting the right technology inside your lab for each type of experiment or project, and you have to weigh all these different factors before you choose which instrument to go with. So I want to show some exemplary data and performance information about Gator to show you how what kind of performance you can expect. This is a CDMO, bioprocessing customer who compared different instrumentation, the Gator system with a traditional BLI platform, and in their hands they see very tight CVs in bioprocess samples. So these are crude samples and purified samples shown on the screen. The CVs that they saw here are very, very tight. So what we're trying to do is really excel with respect to what products we create at Gator in terms of reproducibility and give you something that you can rely on for daily workflow. This is just additional data to show that Gator BLI data compares very favorably with HPLC in quantitation assays. In terms of throughput, how are we solving throughput challenges? We have a variety of systems to scale with your throughput needs, from 4-channel systems up to 32-channel instrumentation. So you have a very high parallel sample processing throughput available on the platform. In addition to the instrumentation itself, we have combined them with automation that is external to the platform so that you can have liquid handling and plate handling delivered to the Gator system. At this show, we're showcasing our collaboration with Hudson Lab Automation. You can see at booth 200, we have the Gator system working together with the Hudson Cyclops robot and other laboratory automation products that they brought into the workflow. So you have the ability to run 24/7 continuous running of your experiments for all of your library samples. There are you can do automation to different levels. You can set up a single batch, or you can have an incubator that has more plates to process in a single walk-away experimentation. So you can really scale your throughput as much as you need with the Gator platform. In addition to just making experiments faster, once you start them, we also want to address the pain point of having sensors that are ready for your experiments at the beginning of the run. So we work very hard to create a wide variety of sensor choices that are off the shelf. So you don't have to really make a sensor with the ligand that you need for your experiment. So in order to make that even easier, we do offer custom sensor preparation. So if you come to us and talk to us about what kind of sensors do you need for your experiments, if we don't have that in our catalog, we'll work very closely with you to make them available on a project-by-project basis. Some of the innovations that we have done on the sensor side are on the chemistry. You can see a large variety here across antibody, protein, and virus particle titer and kinetics analysis. But we also work on improving the sensor performance for different applications. For example, we have about 5 different varieties of streptavidin sensor itself for various applications. The typical streptavidin is very commonly used in broad portfolio of applications. We have a streptactin or a streptavidin XT sensor, which gives you much more signal, like 2 to 3 times more signal than the streptavidin sensor. And that can be useful in peptide and smaller molecule applications. We have SMAP, which is really focused on really small molecule, drug discovery type applications, and so on. So there are more choices depending on the type of application that you wish to do. So we would be happy to work with you on your specific needs more quickly than you may realize. This is just a quick example from a customer to show that the Gator Pro system, the 32-channel system, basically screens a 96-well plate very quickly. Here is another example of a customer who is a CRO that supports protein-protein interaction analysis, measurement for AI and other antibody or protein design libraries generated from AI and other sources. They basically run their wet lab operation based on BLI technology. They have scaled their operation over the last year to 2 years, as they show on the screen here, going from about 3,000 experiments to about 30,000 experiments. This is about 3 different Gator systems in their lab, and they are automating 2 of them. So they want to even scale higher with this. This is just an example of how Gator is used in a setting where a commercial operation relies on its day-to-day availability, the reliability of running it for clients, and being able to build a business on this platform. Then I want to address something in the background, which is just reducing the cost of ownership. Costs are going up very significantly, especially since COVID, right? And it is not all completely worthy of being transferred to the customer. That's at least our take on it and our opinion on it. So what we're trying to do is really reduce the cost that you have to pay to get high quality, reproducible performance in your lab. So generally you'll see that we're about 30 to 40% lower cost of ownership. And we can talk to you a lot more about this as you, if you're interested. In the interest of time, I'm being asked to cut this short. Here are just a couple of examples of customers who are very happy with Gator systems. So we have Yeah, basically 8 out of the top 10 biopharma have Gator systems, so we're not a young company. We're a pretty established company and very reliable partner for you. We have facilities in Palo Alto in California, and in Shanghai and Suzhou. We have about 5 R&D facilities and about 4 manufacturing facilities. And so we are here to work with you and innovate with you to meet your emerging needs. I'll cut it short. Thank you very much for your attention.